Hello, my name is Neil Lagali. I'm a professor of experimental ophthalmology at Linköping University in Sweden. Today I'll be talking about congenital aniridia, focusing on the genotype, the phenotype, and also several different gene therapy possibilities. First, a short overview of congenital aniridia. Congenital aniridia can be caused by any one of over 700 identified mutations in the PAX6 gene. And this gene is a very important gene because it controls the development of the eye in utero. Uh, Mutation is usually confined to a single allele and results in 50% of the normal PAX6 protein production that leads then to the underdevelopment of the eye. Aniridia can arise sporadically or it can be inherited from a parent. It is classified as a rare disease and has a prevalence that varies from country to country, but is approximately 1 in 70,000 individuals. And as I mentioned, there's more than 700 unique mutations in PAX6 that have been reported to cause aniridia phenotype. The phenotype of aniridia is most prominently hypoplasia of the iris. It can be partial or total iris hypoplasia. Also foveal or macular hypoplasia. We don't not seeing the foveal dip or it's underdeveloped. Also, glaucoma is prevalent, as well as cataract development, nystagmus, and a progressive keratopathy. So it's important to mention aniridia, what we call aniridia syndrome. This is because aniridia for a long time was thought to be isolated, meaning can affect only the eyes. However, now it's becoming accepted that aniridia is not only an eye disease, but several other areas of the body or organs are affected. And it's illustrated by this picture here. We can see in the picture that the brain, various areas of the brain can be affected, including the pineal gland. Also, there's an olfactory deficit, audio deficit. The pancreas, which also expresses PAX6, is affected in aniridia, as well as, of course, all the eye, different eye structures from the back to the front of the eye are affected. Importantly, the tissues of the brain and the pancreas do express PAX6, and this is likely why we see changes in the brain and pancreas. This could lead to a number of different systemic conditions, for example, obesity and diabetes. Also, sleep disturbances are widely prevalent, and as well as hearing and smell deficits as well. Next, I would like to spend a little bit of time talking about aniridia-associated keratopathy, or AAK. This is because we have a specific research interest in this area, but not only this, but AAK is one of the progressive changes that occurs in aniridia, and it is one of the pathologies in aniridia that doesn't really have a good therapeutic option or treatment today. For example, the cataract can be treated, the glaucoma can be treated, even the iris defects can be treated. However, eventually it's going to be the keratopathy that's going to be the vision limiting factor in most patients with aniridia. So as I mentioned, it's a progressive, it's a conjunctivalization of the ocular surface. So the conjunctiva starts to grow over the ocular surface in a pannus, and this is believed to be due to insufficiency of the limbal stem cells. So both the limbal stem cell niche is compromised in aniridia, and also the conjunctiva is also abnormally proliferative. So this results in the aniridia-associated keratopathy. Part of the keratopathy could also be considered as being abnormalities in the tear film as well. Also, the tear composition is abnormal, so there is a meibomian gland dysfunction. Also, inflammation is quite obvious and present at the ocular surface in the cornea, as well as in as it progresses in later stages, vessels start to grow into the cornea. And also from a young age, there's often a neurotrophic deficit in the cornea, and this is a deficit of nerves, and that is important to maintain the corneal epithelial homeostasis. The result of all these changes is that the ocular surface is chronically inflamed, it's dry, there's a nerve deficit, and this leads to, for example, poor outcomes for any type of corneal replacement surgery, such as keratoplasty or limbal stem cell transplantation. What often happens is the tissue gets rejected and the transplant fails. So just to show you what this looks like in the clinics with the keratopathy, there's a grading scale that we've developed to be able to grade the severity of the keratopathy, and it's based on the progression of this conjunctival pannus that grows over the cornea over time, and we can see here in the images that in the grade 0 case, which I'll highlight here, there is a clear limbal border and there's no overgrowth into the peripheral cornea. However, in grade 1, the conjunctival tissue starts to breach that barrier and come towards into the cornea about 1 millimeter or less into the cornea. In grade 2, you can see here that the pannus has entered the peripheral and paracentral cornea, but not reached the central cornea. In grades 3 and 4, the central cornea is also compromised, so the visual axis is compromised, and this is overgrowth of vessels and conjunctival tissue. And in grade 4, it's an end stage with thick pannus and a whitish appearance. So it's also important to realize how much the phenotype of the eye that's seen in the clinics is dependent on the genotype, and this is illustrated in this slide by the keratopathy. We know from research that the grade of the keratopathy, which I showed in the previous slide, is strongly related to the type of mutation within PAX6. There are different classes of mutations that are illustrated by this table. From the top row, which is the mildest, and moving down the table to the most severe. So if we look at the mildest non-coding mutations in PAX6 that don't result in a direct impact on the regions of the gene that code for the protein, these kind of mutations occur in about 10% of cases, lead to a very mild phenotype that's not really progressive with age. Also, missense mutations occurring in about 10% of the cases lead to a very mild phenotype and a mild progression. The bulk of the mutations, about 70%, are what's termed premature termination codon or C-terminal extension mutations. And these are the classic aniridia, which is age-dependent and progressive. And finally, we have the most severe, which is an entire deletion of the PAX6 gene and neighboring genes on the chromosome. And this leads to a very severe phenotype at a very young age. And the classic example of this is the Wagner syndrome, where several genes on the chromosome are deleted. So this just goes to illustrate that there's a very strong genotype-phenotype correlation, and that we should really know what the genotype is also in the future for specific targeted gene therapies, which I will be talking about now. So the first gene therapy that I would like to talk about is adalurin. And this adalurin is a drug. It's actually an approved drug. And it's a drug that is called a read-through drug. Now, what this means is that the drug allows the translation of the RNA from PAX6 into a protein because where the mutation is encountered, the drug helps to read through that mutation and produce a full-length protein. So as I mentioned in the previous slide, premature termination codon mutations are the most prevalent with about 70% of cases, and in these mutations, the protein gets truncated and degrades, so we lose PAX6 protein. And these are caused by, for example, nonsense, splice site, and frame shift mutations. So adalurin is a drug that hops over this premature stop codon in the case of in-frame or nonsense mutations. And so theoretically it would allow to produce the full length PAX6 protein and thereby correct the genetic defect. The drug has been approved for use in Duchenne muscular atrophy, but not for use in the eye. However, in 2014, the team of Gregory Evans et al. From Canada applied the drug to a mouse model of aniridia. And here we see in the left slide what the result was with systemic administration of the drug in mice. And so these are cross sections of the eyes. If we look at this is a normal wild type eye, and this is a untreated aniridia eye, and you can see that's very much malformed. The lens doesn't develop, and that the tissues don't develop and separate as they should. Giving the drug adalurin to these mice systemically from postnatal days 4 to 14 for just 10 days resulted in this normalization of the eye structures. So that was systemic administration. On the right side, we see a topical administration to the eye from 2 weeks to 3 months after birth, giving the drug daily as a topical eye drop. And we can see here again that on the right side here, we can see that the gene therapy has resulted in a more normalized eye structure compared with the non-treated aniridia mice. So this is a very exciting result because it's already an improved drug for a different indication. So what happened to the cornea in that study? They treated the mouse cornea for 60 days with adalurin. And what we can see here is comparing, they did a treatment systemically, but also topically. And we can see here, systemically did not rescue the epithelial phenotype. It's a very thin and fragile epithelium in the aniridia mouse. However, treating topically for 60 days did restore the multi-layered epithelial phenotype. And this is very important for preventing development of the keratopathy. So in the mouse model anyways, the topical treatment did restore the corneal epithelium. So what happened after these positive results, first results in the mouse model? Well, adalurin was then taken into a clinical trial in the United States and Canada for patients with aniridia. And this was registered on the clinicaltrials.gov website here. And the study was a study for over 2 years, taking the oral drug, so it was delivered orally 3 times daily, or some patients received a placebo for 1 year and even up to 3 years. The main outcome of the study, however, was a change in reading speed in patients who are over 8 years of age. So this is not this is a functional outcome, but it's not a direct objective outcome in terms of the phenotype. Patients in the study were not selected for severity of aniridia. There was a wide range of ages of patients included. and the results, perhaps not surprisingly, was that there was no effect on the reading speed. And due to this negative result, further trials with the drug were cancelled. There's been some discussion about this, and perhaps that instead the topical Adalimumab perhaps should have been given to a carefully selected group of aniridia subjects. However, at the present, there are no trials planned. Adalimumab, however, is not the only read-through drug. There are other read-through drugs. For example, amlexinox is another such drug. And in a recent research study, this was applied to induced pluripotent stem cells that were used to derive both corneal limbal stem cells and retina models. And this was done in this paper shown here in a group in the Netherlands. And the idea here was that cells were taken from aniridia patients and then reprogrammed to induced pluripotent stem cells, which can then differentiate to virtually any cell in the body. So the team then derived corneal stem cells, limbal stem cells, and retinal organoids from these patient-derived iPSCs. Then in vitro, those were treated with the drug amlexinox, and the team showed that the drug could restore the level of PAX6 protein to normal levels in these corneal stem cells and in the retinal organoids. And those cells and organoids then also expressed other protein markers that were markers of normal tissue. So this picture shows the sort of concept that taking cells from aniridia and controls and then generating iPSCs, which are then used to generate retinal organoids or corneal limbal stem cells, and then those are treated with the drug to show that the drug could normalize PAX6 expression and expression of other markers. So this is quite an interesting study showing that other read-through drugs like amlexinox have the potential to correct the lower production of PAX6 protein. However, thus far, this drug has not yet been taken into animal studies. That would be sort of the next phase that would be needed to show that this actually works in vivo in a living organism. Another class of interesting drugs that have come up recently are the MEK inhibitors. And the MEK inhibitors are actually a class of cancer drugs that are currently in clinical trials, and they target the MEK-ERK pathways in the cell. It's known that the MEK-ERK pathway, and especially its suppression, will increase the production of PAX6 in cells that normally produce PAX6. So these MEK inhibitors have been in recent years applied to the mouse model of aniridia, and here's a paper from 2020 showing this. And this group from Chicago showed that the MEK inhibitors were able to increase PAX6 protein levels in vitro in human corneal epithelial cells, and they could increase the PAX6 protein level by 1.5 to 2 times. So theoretically, then compensating for the genetic defect. And here's the graph to show that. In the same study, the investigators showed that using an eye drop solution of the drug applied twice a day from postnatal day 5 to 30 in the mice could rescue the phenotype change in the aniridia mouse. So they applied the treatment, 5 days of treatment, and then 2 days without treatment to minimize potential toxicity of the drug in the eye. And then they showed, as you can see in the image here, that compared to just giving the vehicle, giving the drug, resulted in a much more open, you can see here, an open eye that was much more transparent, as well as the structures of the eye had a more normal appearance, and importantly, looking at the cornea, its epithelium had a normal appearance, a multi-layered epithelium, whereas it was clearly hypoplasic in the aniridia case. They also showed here that the drug increased the level of PAX6 production compared to the non-treated, as well as the marker keratin 12 in the corneal epithelium. So these are promising results, and the next steps are to use this drug in a more relevant model where the aniridia develops more slowly in mice and is not as aggressive as this model to mimic the human aniridia. And also they would have to develop a proper dosing regime as well. It can't be given, for example, from postnatal day 5 to 30. Would have to be tailor-made for human patients. Another interesting drug that has come up in recent years is duloxetine. Duloxetine is already an approved drug. It's a generic antidepressive drug. First identified by Averdam and colleagues in France from a drug screen. And they published this study that is shown here, and it was an in vitro study in limbal stem cells of the cornea that were mutated using the CRISPR-Cas9 system to knock down PAX6 expression in one of the alleles, so thereby mimicking aniridia. And what the drug does is it suppresses this MEK-ERK pathway, just like the MEK inhibitors, and by suppressing the MEK-ERK pathway, it increases the level of PAX6 protein production, as can be seen in this Western blot diagram here. Interestingly, and unlike the MEK inhibitors, the duloxetine works on the wild type allele and not the mutated allele. So it thereby overproduces PAX6 on the normal copy of the gene. So it doesn't depend on the mutated copy of the gene. So in that way, it would work for any type of mutation, not just the premature stop codon mutations. Also, it's a cheap generic drug, and so it has advantages over MEK inhibitors, which are not really approved treatments yet. There's a lot of toxicity and safety information on duloxetine that we don't have with the MEK inhibitors. So the plan is to now test this drug in aniridia mouse models to see if it can rescue the phenotype in the mouse models. Another interesting gene therapy approach is gene or genome editing, and this is become famous because of the CRISPR-Cas9 gene editing technology. And so it's using this technology to correct the genetic mutation that causes aniridia. Right now, we know there are clinical trials ongoing for other diseases using the CRISPR-Cas9 genome editing technology. And the group of Simpson et al. In Vancouver has tested this in mouse models. So what they did was they used lipid nanoparticles, which are small particles, to deliver the components of the CRISPR Cas9 system to transfect cells, either ex vivo or even in vivo in mice, and thereby have the gene editing occur in the living cells or in the living tissue. And what they showed was that this could actually work in cells, and also that this vector, the lipid nanoparticles, could be delivered, for example, to the cornea. In that same study, where they showed in vivo genome editing with the CRISPR-Cas9 system. They could show that the exact mutation in terms of the base here could be corrected, for example, from a T to a G with this lipid nanoparticle CRISPR delivery system. So this is very precise editing of the exact mutation, and this could be done in vitro. However, when they tried to inject the LNPs into the cornea, they did this intrastromally in the mouse cornea, and what they found was the red signal here shows where the actual gene editing occurred. So they did confirm that the gene editing can occur in the cornea. However, this signal was only in the stromal and the endothelial cells of the cornea. So somehow the gene therapy was not transfected to the epithelium, which is, of course, where PAX6 is expressed and which is the most important cell type in the cornea for potentially preventing the keratopathy. So at least this was a partial success with the gene editing. However, more work needs to be done to see how best to deliver this to the corneal epithelium. And finally, perhaps something that's coming in the hopefully in the near future, but it is technically possible today. This already exists. And this is prenatal therapy. So this is called pre-implantation in vitro fertilization screening. And the concept of this is that if a patient with aniridia wants to have children, normally the risk for inheritance is 50% to any offspring. However, there is an option instead of a normal fertilization to have in vitro fertilization. And this then makes the embryo available for genetic examination and testing before it's implanted back into the mother. And this kind of a technique would work for monogenetic disorders like aniridia, which is principally in the PAX6 gene where the mutation lies. So it's called pre-genetic testing and monogenic. The way that this works is that while the in vitro fertilization procedure is being done, at the point where it's day 3 of the in vitro embryo development, one would take a small 2-cell biopsy. At that point, at day 3, the embryo is only 6 to 8 cells large, and you would take 2 cells of those. And or another option could be to wait until day 5 when the embryo has anywhere from 80 to 100 cells, and then take a 6 to 8 cell biopsy. In either case, once the biopsy is taken, the cells are then sequenced, and then by this means you could determine whether that embryo actually harbored the mutation or not. And in this way, you could select the embryos that don't have the mutation for the in vitro implantation back into the mother. And that would then result in the development of a normal baby without the mutation. There is, this is not 100% foolproof. There is some chance for miscarriage, but the success rates can be up to 85%. And the great advantage of this is it could allow couples certainty in not passing the disease forward to future generations when one of the parents has aniridia. However, on the downside, it could be quite an expensive procedure because in vitro fertilization is generally expensive, and this would require an additional screening step and sequencing step as well. However, it is feasible, and it has been applied, for example, to diseases like Huntington's disease. It hasn't been yet applied to aniridia, but this is just probably a matter of time. And with that, I would like to conclude the talk and thank you very much for your attention. And I would also like to thank our funders, the Restore Vision EU project, as well as the Cost Action Aniridia Net.eu. Thank you very much for your attention.